Good morning, everyone. In the last class, we were discussing about cancer stem cells. We discussed about their properties and how to experimentally determine them or characterize something as cancer stem cells. If you quickly remember, the cancer stem cells have three properties. One of it is self-renewal, and the second one is tumorigenicity, ability to produce tumors, and the third one is heterogeneity, ability to differentiate. So if you look at self-renewal and ability to differentiate into something else or cause heterogeneity through asymmetric cell division, they are properties of stem cells as well. So what is additional here is the ability to form tumors. Okay. And of course, the source of these stem cells, the cancer stem cells, is cancers. They take biopsies of tumors and then they will try to culture and so on. So in that process, to prove each of these properties, we have discussed about several techniques involving FACS and other things, serial transplantation, in vitro methods, in vivo methods, and so on, right? Tumor induction can be shown in vivo using SCB mice. Self-renewal can be shown by serial transplantation. What we have done here, tumor induction, that can be kept on done repeatedly. Take a tumor sample, put it into mouse, take the tumor of it, take a small bit of it and put it in a new mouse and so on. That is serial transplantation. And then we have spear formation, that is something similar to that of embryoid bodies we have discussed with embryonic stem cells. So cancer stem cells have a property that if you take one cell or group of cells and culture them, they can form spears like this. Those are called as spear culture. If you remember, when we were discussing about the hanging drop method, there were nice new technologies where there are pores and this is the hanging drop, something like this, and cells can clump together and form spears. In that location, we discussed it with regard to embryoid bodies, but similar techniques can also be used for the spear formation. We call it a spear here because it is forming from cells that belong to the cancer. And then we have tumor heterogeneity that can be proved by FACS. That means if you analyze it by flow cytometry in terms of forward scatter or and side scatter and so on, we can distinguish that there are at least more than one cell type, right? That means something in cancer stem cells can give rise to more than one cell type. One of it will be, of course, self-renewal. That means it will form a cancer stem cell, but it can also form differentiated cells, some other differentiated cells. Or you can say, we cannot say, when we use the differentiation, we use it as a normal in developmental biology, we would use it for the production of a certain type of cell, functional cell. In cancers, these are all abnormal. So we might not want to use the word differentiation here, but still, I hope you get the point. They might be non-functional, but they are different from that of the cancer stem cells in terms of morphology and maybe function as well. So in that, we take this solid tumor, make a single cell suspension, go for cell sorting. We might get two different cell types, cell type A and cell type B, and each of them can be given to different mice, and we can find out if they are forming a tumor or not. If they're forming a tumor, then they are likely cancer stem cells, whichever forms. So we also discussed about the minimum number or the lowest number of cells required for the formation of the tumor. I hope you remember. And also brought into light that the site of injection where we are putting in the cells. Not all cancers can grow everywhere or anywhere in the part in the mouse body. So we need to check different of them, and the ability with which they grow in different tissues is also varying. And another point I brought is about the lowest number required. If there is lowest number required is less, which would mean that the proportion of cancer stem cells in that tumor is high. Okay, that's the meaning of it. So in vitro assays, spear formation that we have just now reviewed, and then there is first identification of cancer stem cells came through cytometry using highest dye efflux, and we discussed about the ABCG2 efflux pump, and we are going to have a quick discussion on that as well. ATP binding cassette. I hope you remember in your cell biology, you must have learned it. These they bind to ATP somewhat like this, as shown here, and then they actively pump out some of the compounds out into the extracellular side. So the highest dye that we're using here can penetrate through the cell membrane into the cytoplasm. If the cell has, assume there are two different cells, one of it has the pump and the other one does not have the pump. So the dye molecules that we are putting in, they will pass through the cell membrane and they will be here in a cell that does not have efflux pump. In other ones, what may happen is when the dye may pass in, but then the dye could be pumped out by these flux pumps. So at the end, when we use Hoy's dye, some of the cells will be stained with the Hoy's dye, some will remain unstained because of the efflux pump. The stain is sent out of the cell. So by doing that, if we performed, made a single cell suspension, and then we have run the flow cytometry based on Hoy's dye, we can get, in this case, say there are quite different cell types. This is one cell type. This is probably another cell type. Maybe here, very few numbers are there. And here is another one that is referred to as the site population. So researchers have tried each of them and saw if they are giving rise to tumors or not. These cells did not give rise to, did not have tumorigenicity capacity. These did not have tumorigenicity capacity, whereas site population ones had the tumorigenicity capacity, which means they are likely the cancer stem cells. I hope you remember that. Why are they stained differently? What we are knowing here is the SP cells are likely the cancer stem cells, but that's not the entire story of it. What we mean is here we are using a dye, and the staining is because of ABCG2, and we are collecting those unstained ones. That means the cells that have expressed ABCG2 are likely the cancer stem cells. That's the question. The next question would be, has ABCG anything to do with cancer stem cells? Or is it a property that, the gene expression of ABCG is essential to be or behave like a cancer stem cell? These are some of the questions. So what people have figured is that ABCG alone may not identify cancer stem cells in all tumor types. In different tumors, different things may be there. As I said, this assay using Hoy's dye cannot be used for all types of tumors, or it's not the same way these cells may be as a stained or unstained. So what it indicates is having high expression of ABCG is not directly related to being a cancer stem cell. That is one of the indications they have it. What they have also done is they have deleted some of the from cells, from the genome of those cells. They made homozygous ABCG minus. When I say homozygous, there are two copies of ABCG2, right? And both these are removed, and so you can call it as ABCG2 2 minus, or minus 2, sorry. That means both the copies are deleted. Even after deleting these genes, cells were still able to form tumors. That means the first indication is even though ABCG is deleted, the cells are able to still form tumors. So these cells are cancer stem cells, right? Because they're able to form tumors, and other properties of self-renewal and other things are also being exhibited. That means ABCG is not directly involved as a essential property of cancer stem cells. That's the meaning of it. So I hope you understood that there is site population is still has different cell types. That means there are a mixture of cells. All of them, the only difference in them is, or what is common in them is probably they're all unstained. And why are they unstained? It is because of expression of ABCG2. So site population is still heterogeneous and probably is enriched for cancer stem cells, but not all the cells in this site population are cancer stem cells. Okay. You don't. Why we are discussing this is the way we can deduce a function. How do we relate a function with a gene? Here we are talking about a cell being as a cancer stem cells. The indication we have at the genetic level is ABCG2. Is there any relation? Is it the cause? Is ABCG2 the cause for a consequence of a phenotype, something like cancer stem cell? That is what we are trying to discuss. And always um, how we relate the causation, the relationship between cause and consequence. It is something, if you want to speak in very trivial ways. Say, for example, a person has fallen down, right? A person has fallen down. And the reason for that person falling down, if somebody else said, it is because in the morning you have seen somebody's face, and that's why you have fallen down. What is the cause and consequence? Can you keep it? Is that logical? Or you can also bring it up that a cat passed through our path in the morning. That's why we have fallen down here. So there could be reasons, there could not be reasons, but how you associate a cause with a consequence is very important in general thinking. And especially in science, the idea is to be as irrefutable. That means you should have tested the cause and the consequence, the link, and always come up with the correct or most appropriate cause for a consequence. It's like for a gene for a phenotype. Okay, many a times there are multiple reasons. Maybe this person has missed a rock and then why he fell, or the person was dehydrated, that's why he fell, he or she, and many things might be reasons. So finding out the correct and most appropriate reason is what is science. Okay. So over the years, people have figured, um, people have tried to taken tumors, different types of tumors, and then they try to isolate cancer stem cells. Remember this, we don't know how to, first thing, from tumors to getting single cell suspension is easy, right? You have techniques, and you can do that. From single cell suspension, we need to do sorting. Based on what shall we sort? We are going to use facts, and Hoy's dye is not is only specific for the ABCG2 producing cells. It is not specific for cancer stem cells. So we know that different cells have different cell surface markers, right? They can be of different shape. I hope you know from biology cluster designation CD44 or CD133, CD24. These are some of the cell surface markers that are present on cells. So people have decided that, okay, if we have a different types of cells, okay, and this is a heterogeneous cell mixture that we have isolated from the single cell suspension, and if we know their cell surface markers, based on the cell surface markers, we could separate them through using facts. Okay, I'll try to use this space here, and let's see how we get it. So I'm not going to write these self surface markers, but I'm going to put a number in between them. And based on that, we will try to, we should assume that, say, for example, I put 44, it would mean it is CD44. And I would use CD24 and CD133. These are the most common ones that are found in majority of the cancer stem cells, in different types of cancer stem cells. We'll get to that in a while. So if I said 44, that means the cell is expressing only cell surface marker CD44. If I said 44 and 24, or 44 and 133, and probably I will use if you excuse me, I'll try to use a different one so it's easy to write for me. Okay, so let's see how to do facts and separate the cells based on cell surface markers. Okay, I have some, say, 7 cells, and this would mean 44, and I will say 24. This is only 44. 44 and 133. Remember, I said CD44, CD133, and CD24 are assumed theoretically. These are the cell surface markers, and based on which we can separate them into separate or sort them based on the cell surface markers they have. And 133, 24, and 133, 133 and 44, 44, 24, and 133, 24. Okay, I'll also give the names to these cells so that you should help me which all cells will form into categories A, B, C, D, E, F, and G. Now, the first thing I can do is I can separate. I want to run facts. So I'm going to use, these are already made into single cell suspension. And now I'm going to go for the next step, that is labeling. Labeling with antibodies. Antibodies conjugated to fluorophores or fluorescent dyes. Okay, so I'm going to use anti-44. That means anti-44, all the cells that have antibody 44 will be will come into one tube, and into the other tube, the other cells that do not express 44 will come. So which all cells will come into CD44? This is plus, we will say. That means they have CD44 expression, CD44 minus. Can you help me which all cells will come in here? Just the numbers. Look at there. So A will come. Yes. Anybody else, please help me out.
So B, C,
B, C,
D, and G.
D, and G. That's right. Which all will go into CD44 minus?
E and F, sir.
Yes. E and F. Okay. So we have first separated them based on CD44 only. Then we are going to go for another round of facts. So each of them separately, of course. Okay, this one, the CD44 plus, we are going to go first with. I'm going to use anti 133. What will I? Which all cells will I get into category? Yes, anybody? Please help me.
So B, C, and D.
So 1 second. 133 plus and CD 133 minus. Okay, tell me now. Which one will go into plus?
So B, C, and D.
B, C, and D. Okay, thank you. And into CD 133 minus?
E and F, sir.
No, no. Out of this only.
So,
Yeah.
okay, sir.
A and G. Is that right?
Yes.
A and G. And what about the other category here? Also, I'm going to do anti 133. This is separate reaction, okay? So in this case, I would have, so CD 133 plus and CD 133 minus. Which cells will go E and, okay, both of them, E and F, both have 133, right? E and, sorry, E and F. So again, I want to do something else. I mean, this is how the sequence of reactions happen. Now I'm going to use after facts. I'm taking this population, and then I'm going to do anti 24. Okay, I'm going to add antibody against CD24, and then I should get two different populations. Which one is BC? Sorry, CD24 plus and CD24 minus. So out of BCD, B, C, and D, none of them have, all of them, B, C, D come in this category, and there are no minus 24. In this one, in the other CD minus 133 population, if
So
you
in
check,
BCD there are no 24 present, no, sir. Then it will come
oh
inside
yeah, yeah,
20.
yes, yes, you're right, you're right. So they'll go into the other one, not here. Yes. So B, thanks. B, C, D are all minus CD minus CD24 minus. Yes. In this population, in the second one, AG, we have A is 24. Okay. I'll put this. This is CD24 plus, and this is CD24 minus. A is 24 plus, and G is also 24 plus, right? And we do not have anything here. So finally, I would want you to tell me what is the cluster designation or cell surface markers of this one, the one that I'm going to point now in this one. Where did it come from? First thing is, I will erase one of it. So for ease, anyway, this is not there. So we'll just go ahead with this. So these cells that I'm pointing at here, BCD, BCDR, CD44 plus, because first we have done with that. Then they are CD133 plus, because again there are plus here, and here they are CD24 minus. This is how we can separate. We are not yet done. We'll just go ahead a little bit more and look at these cells here. So they would have been CD44 plus, then 133 CD133 minus, and they are CD24 plus. Okay. If we have, so usually what they do, something like this is, they would have made a single cell suspension, like in this case here. Okay. And then they would test whether which of these cells are forming tumors. And for our simple understanding, we would assume that, say, CD44 has the ability to form tumors. In that case, these cells might be discarded. Then they are going to do based on CD133, okay, by using anti-133 antibodies. And then sorting is done. We got a population expressing CD133 and a population not expressing CD133. Again, they would have checked for which of these two is causing tumors. Okay. And then they might find that, say, this one, the ones that are expressing 133 are forming tumors. In that case, these cells might be discarded. Okay. Now we know that the cancer stem cells so far are expressing CD44 plus and CD133. We don't know about about CD24. Then they would have separated based on 24 and see whether which of them are expressing, which of them have the tumorigenicity capacity or cancer stem cell properties. And they would have narrowed down to something like this. So this goes with a bit of trial and error. We don't know what is present, and then if we don't know and still we have to go ahead, then this is how people will proceed. Okay. Any questions in this part? So this is how the experiments are usually done. Say majority of the cases we'll find CD133 and CD44. It's a prominent one. I don't need you to mug up this, but the technique part is what you should actually know it. It's a transmembrane prominent one. It's a transmembrane glycoprotein that has some functions. Similarly, side of CD44, it's a receptor for hyaluronan, extracellular component, and then it is involved in cell signaling and so on. So CD44 is also found in majority of the cancer stem cells, isolated from different types of tumors. Here is a table which summarizes the what is understood or found so far. And I want you to look at the different cells, the markers that are present here. Just give me a minute. So here we have for breast cancer, this is CD44 plus and CD24 minus. I hope you understand how they got to this level, right? By a series of experiments or series of facts based on cell surface markers. And here, this is fraction. Percentage means somewhere between 11 to 35% of the tumor, of breast cancer tumor, consists of cancer stem cells. Okay. You should also remember here there are only 2 of them given. You can still refine them using many more other antibodies against several other cell surface proteins, and probably we'll be able to isolate exactly which of these properties define cancer stem cells, or at least the cell surface markers that are specific to the cancer stem cells. This is important, and we'll discuss that why in a while. So in brain tumors, they found those cells that have expression of CD133 plus. They are likely the cancer stem cells, and they make up about 5 to 30% of the fraction of the tumor cells. So I'll just take a little bit of this one, and the other ones you should know, or at least what you should know is CD133 and CD44. You will find them quite often. 133 is here, 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 and here, and here. 44, there is CD44 is something else which is found quite often as well. But there are also tumors where you don't find CD133 or CD44. So it depends on the tissue, it depends on the tumor, and several other things. I'll explain this one so we will know what it means. I hope you remember what is alpha 2, beta 1. Those are integrins that are on the cell surface. They are made up of two chains, alpha chain and beta chains. Alpha chains, you have different types of alpha chains, 1, 2, 3, like that. And beta chains, you have again 1, 2, 3, so many of them. So combination of integrins could be on cell surface markers. At least in this case of prostate cancer stem cells, they found that those cells that have alpha 2, alpha 2 beta 1 integrin, if it is high in concentration or say numbers on the cell surface, they're likely they behaved as cancer stem cells. So that is one other cell surface markers. So why we are talking about these cell surface markers and why did we spend time on this? It is about our objective is to kill the cancer stem cells. In all of our course, we are trying to make regeneration or growth of the stem cells and differentiate and allow the individual to survive. In cancer stem cells, we are studying it so that we can kill them, so that the cancer we will be able to treat cancer in the future. That's the objective of why we are studying this here. So if we want to kill the cancers, we want to target the cancer stem cells. How can we target the cancer stem cells? We can only target them if we know they are resistant to radiotherapy, they are resistant to chemotherapy because they are usually kind of dormant. They will only divide when it's required, and they may have additional protective areas, something because of, say for example, abcg2. If you're using a drug to send it to kill this cell, the abcg2 can pull or affects the drug out of the cell, thereby protecting the cancer stem cell. That is another problem. Firstly, if something is very dormant, you do not they are usually resistant to majority of the types of damages. Those with high metabolism or high rate of cell division, they are the ones usually they are vulnerable to targets. But cancer stem cells seem to be very resistant to these kinds of um, uh, the drugs that we try to do. They are resistant to, because of dormancy, they are also resistant to radiotherapy and so on. And maybe there are many other mechanisms because of which these cancer stem cells remain quiescent, or efflux pumps pushing out the drugs that we are putting. And in that case, how can we specifically attack cancer stem cells? The idea would be something like this. So if we have some cells and some of the tumor cells, assume we have gotten some tumor cells, and out of which some of them are cancer stem cells. I will just put this as cancer stem cell. And each of them have their own cell surface markers. Say, for example, I'll just put it as, okay, I'll put cell surface markers CD 44 and 133. We have cell surface markers 133 in other cells. Some cells have 44 alone, some cells, yeah, 44 and 133. Now, can you think of a way where if none of the cells had 44 or 33, it would be easy to target them, because we will have a lesson on how to deal with these. But right now, just as a general idea, you can have antibodies linked. This is this with a toxin or some radionuclide and so on, doing something like this or making different other types of antibodies is antibody engineering. So you might also want to look up this when you're studying immunology. It's a very interesting one. Okay, I hope you know. Have you heard the word abzymes? I heard it long time ago. We know about enzymes. We know, I hope you remember about ribozymes. What are ribozymes? Yes? Anybody? Try. If you
So
miss it,
it is an RNA enzyme.
yes, RNA molecules that can act as enzymes. We studied them in introns and other things. Enzymes are antibodies that can act as enzymes, right? They are also this is not exactly related to what we are discussing, but as part of antibody engineering, probably you can also think about enzymes. Anyway, antibody engineering is, I hope you might be studying. The dotted area is the hypervariable region, or that is where complementary determining regions are present. Those are the ones that recognize the epitopes. In this case, what is the epitope? Some part of the cell surface marker of CD133 would be the epitope. So the antibody is raised against, say, for example, anti-CD133. If I said anti-CD133 antibodies, that means those are antibodies that can recognize specifically the CD133s. And the region where the toxin is bound is the constant region, heavy chains. And there you can engineer to conjugate a toxin or a radionuclide or something like that, so that they can be, the antibodies are likely to bind here. They might bind here, and they might be internalized. Okay. And then the toxin will be delivered there, and cancer stem cell would be killed. Why are we targeting cancer stem cell is simple and straightforward. We want to kill those cancer stem cells so that the if we in the conventional therapy we are killing the all the other cells but not the cancer stem cells, so the tumor relapse is occurring, or tumor relapses. Whereas if we can somehow specifically target the cancer stem cells, then if we have killed it, the rest of the cells would die out by wear and tear. Okay. So the goal is therefore how to target cancer stem cells. We are a long way from this. We are not too close to find that, because there are issues with trying to identify it. The first one is identifying the cell surface markers. Right? That is the first one. If we said CD133 is there, CD44 is there, like in the previous slide, CD133 is also present on other cells. It's a normal gene product, and it has some functions in the normal cells. And if we are using anti CD133 antibodies, then we are also likely to kill the normal cells as well. That will be side effects. Most of the treatments, majority of the treatments will have some degree of side effects. And this is, people are very scared about it. I am too, about the scared effects. I mean, we are scared of side effects. So some of the companies will say, here is a medication without side effects. And is that true or not? If there are no side effects, are there any effects at all? That's a question. If we take anything that we are supposed not to take, or we don't take normally, is likely to have side effects, at least on the continued usage of that medicine. So there is something that doesn't have, probably a medication that doesn't have side effects is food that we eat, right? Nutritious food, delicious food. It can influence our mind or brain, and then some kind of placebo effect, and put our mood up, and then we can probably heal some of the things. But many times when companies sell as medication without side effect, or natural products no side effects, we have to be very suspicious about the activity or effectivity of that medicine. Okay, so this is, in general, this is how you have to think. We don't take everything just for granted because somebody said it. I'm not saying what should be taken, what should not be taken. I'm only saying how you can probably, or what you should pay attention to when you are when you're taking a decision about, or forming an opinion or notion about something, whether a particular thing is a medicine or not. Okay, I as a matter of teaching, this is, or in general also, we don't need knowledge. I don't need to deliver you the knowledge, or at least I should put it as, I should give you the skills to sift or decipher the knowledge or derive the knowledge. This is my job. Okay, or I can put it, I have to give you the knowledge of the skills, right, so that you can derive your own knowledge. So think about these things, don't stop thinking. And if you are directly getting knowledge without knowing how it is derived, then we might be making some mistakes, because if the person who is giving you that made some mistake, then you are making that person's mistake too. We can make our own mistakes, not of others' mistakes. Okay, so I hope you understood why we discussed about cell surface markers, and the goal is to kill the cancer stem cells. The risks are that normal cells also have these markers, and in that case, how do we specifically find out? The ideal goal for cancer stem cell therapy is, the first one is to identify specific cell surface markers of cancer stem cells. That is one we want. We want to identify that is expressed in cancer stem cells, but not in any other cells. That's a little ambitious too. The second one is how to kill the cell or the cancer stem cell. We will have a lesson on it, different strategies that are there. We will try to discuss there in more detail, but right now we can use toxic compounds, toxic proteins, like in the diphtheria toxin, or we can use radiotherapy, where specifically, if you can figure out these cell surface markers. We could have had antibodies with radio elements conjugated to them, and when it is delivered into the body, it will be internalized by the cells that have the epitopes for this antibody, and those cells could be killed. And that is another way. So far, there has not been absolute success in this. Okay, but the goal is to treat cancer, and if you want to do that, that is actually very nice. So these are the two problems. Just for the sake of discussion, I would want to discuss about. Just give me a minute. I want to have a discussion, and it's about how we can actually feel about it in terms of cancer therapies. I remember one Facebook post, and people just simply say, how is that you are all scientists working and so on, and you're not able to find a vaccine for cancer or vaccine or treat cancer. Cancer is a complex disease. It is not one disease, right? It is the only common property, as we discussed earlier, is that the only common property is dysregulated cell cycle. The rest of the things are different, and it's not easy to treat any of the cancers. I'll try to post it when I find it. And it's actually an important way of trying to see what is right and what is wrong. Because the whole information that we get is um, full of information, and not all of it is true. And we should always be, at least because we are in science, we are supposed to take a different stance, verify the evidence, look into properly, because we can understand it. If we didn't understand, then it is a different issue. But if we can understand, and in that case, we should take efforts and try to verify the available evidence with the available knowledge and not be lazy about it. Usually, when we directly go from here to the information directly, the knowledge without verification, that is when the knowledge that we have could also be contaminated with so much of false information. So if you are improving the skills to how to figure out which one is correct answer, how to verify the evidence that is provided, the scientific method, then probably you will have the skills to know, verify the information. That whatever is coming into you, whatever you believe is right or wrong. So especially with the COVID thing, everybody proposes a different kinds of medicine, and we cannot go by anecdotes. We have to go by statistics, and that's why if you are planning to be in science, and this is something very important for you, there are people who are doing just a PhD in statistics. In the future, all of our jobs are about data management. How much data, how critical data, how many facets of data can you handle and sift and get through the correct required information. And in that sense, being able to do statistics is an important skill that you would require for any kind of information that you will get. So keep that in mind and try to know. I think you're having statistics now, numerical methods and other stuff. I don't know. What do you have in Maths 5 now?
Yes, sir, we have statistics.
Yes. If you have it right now, please do it very, very well. You also have some programming related to it, R program and so on. You can learn it through Coursera, and that will be very important. Okay. Any questions? in cancer stem cells. We will discuss 3 or 4 different types of cancers. There we will go specifically about prostate cancer, breast cancer, melanoma, and maybe leukemia. Okay? Any questions?